Hello and welcome to Cal Week 2020. My name is William Hou and I'm a third-year undergraduate student studying bioengineering with a concentration of cell and tissue engineering. I've been conducting age-related research in the campus lab for almost 2 years. And in this presentation, I'll briefly go over some of our exciting findings and future directions. As we all know, human beings age like the other organisms on Earth. And for many years, people have envisioned that they can reverse this process, either with the fountain of youth or the elixir of life. The reality is, we have not found any of these mythical items. But the good news is, we have identified some candidates. Here's a famous experiment conducted by the Combat Lab a few years back. Basically, we exchanged the blood between old and young mice. And we discovered that there is something in the young blood that makes the old mice younger in terms of tissue regeneration. One of the key factors that we discover is oxytocin, a neuropeptide that promotes trust and bonding between humans. Turns out, oxytocin also plays an important role in regenerating tissues. Take a look at the graph on the right. We injured the mouse muscle and injected oxytocin afterwards to see if it stimulates muscle to grow back. YOTA is young mice injected with oxytocin antagonist, which is a molecule that blocks oxytocin activity. YOT is young with oxytocin only. YV is young with vehicle, which serves as a control group. OV is another control group, which stands for old with vehicle. And the furthest right is old with oxytocin. After a few days, we measured the regeneration based on how big the muscle would grow, and we detected a large increase between old mice with oxytocin and old mice without, as we can see in the left bar chart. The right bar chart measures the difference between the percentage of the injury area occupied by the connected tissues. We can also see that old mice with oxytocin has a drop in such a percentage. Isn't it fascinating? Wait, there's more. ALK5 inhibitor is a molecule that blocks the TGF-beta pathway, which are reported to cause inflammation with the signal intensity is high, when the signal intensity is high. By blocking the signal, we detected higher proliferating and self-renewal signals in both the muscle tissues and neurons, as demonstrated in graph B and C. Now, here's the million-dollar question. What if we combine these two treatments? Will it enhance the regeneration process? Will it become worse? More importantly, what protein levels go up because of the treatment and what go down? To answer these questions, my team and I are currently working on using BONCAT technology to label and quantify newly synthesized proteins. BONCAT uses A and L, which mimics the properties of a methionine and amino acid, so that it can be incorporated during protein synthesis. After we injected the mouse with OT and/or ALK5 inhibitor, we can detect the fluorescent signal from the A and L tagged biotin clicked complex with an antibody array in graph B. We don't know what we will find yet, but it is solving for the unknowns that makes scientific research a marvelous and extraordinary journey. Here, I want to thank my mentor, Dr. Chow, and the individuals in my lab for the brilliant work. My PI, Professor Irina Comboy, and the Department of Bioengineering for offering me this awesome opportunity to present my work. You can click the link below for the published paper I went over. Thank you for listening, and if you have any questions, please feel free to contact me through the email or connect me on LinkedIn. Stay safe and go Bears.